So, dear colleagues, it's my greatest pleasure to introduce you the Austrian partner of Dr. Zinovieва, and it's my greatest pleasure to see in person Professor Johann Sölkner from Vienna University of Environmental Resources and Life Sciences. And here we are going to talk about a European view on this very nice, successful collaboration. Hans, please.
Thank you very much. Can you hear me well?
Very nicely.
It's okay. Okay, so yeah, I put this title up, but mainly I will try to follow the process that you asked us to do, looking into how to collaborate and how to run labs and things like this. And so what I'm writing is fact, not fake, for genomic revolution in livestock breeding. I will give you roughly a short background about my person and about livestock breeding, and then the main issue is the question on, well, my topic at heart. I'm working on genomic selection actually, and then how is my lab linked to the breeders? That's coming up back to exactly what Natalia just discussed with you. And my personal feeling is that international collaboration is kind of, at least it's my key success of success, and I think it can be a key of success for many groups actually. So rather than having years, I tell a bit about myself. So I was growing up on a dairy farm, and I find this one very useful because I understand how breeders are thinking. So that is useful. But when I was working, well, as a child, my father asked me to work with him, I was not interested in his fable for tractors. I was much more interested in the data of the cows. And this, well, continued from there. So I studied at the university where I'm now a professor at, graduated there, and then got professor at some time, which is not very usual in well, Austria, Germany, this area, where typically they don't want to have a professor from their own university. So this, you have to have a very convincing argument to get professor at your own university. Well, later on, after I already had been professor, I was invited to be a visiting professor for molecular animal breeding at the University of Sydney in Australia. And from there, I brought a project back to Austria on genomic selection, and we implemented genomic selection in the Simmental cattle breed very early on. And another thing that I'm very interested in, and where I like the data that we have and the way we can use them, is using genomic data to understanding diversity aspects. Again, coming back to what Natalia was telling. So for animal breeding, I have here kind of a hall of fame. So as an animal breeder, you have to define what you want to have. So defining the breeding objective is key. And once you are clear what you want to have to keep records that relate to the breeding objective. And then what people do is they estimate breeding values. I will go into this one, how this one is being done in one of the next slides. But essentially, so you find out which are the best animals, or you try to find out which are the best animals based on records, and then you mate the best with the best. But the phenotypes are the key for this one. So even in times of genomics, if we don't have proper phenotypes, a lot of phenotypes, they are kind of useless in terms of breeding. They're useless. They're useful in diversity studies, but not in terms of breeding. So you always have to link it to phenotypes. So traditionally, this is a Simmental breed. That's the breed my parents had at home, for example. It's the most popular breed in Austria. 80% of cattle in Austria are Simmental. And so what used to be done is people were mating the best cows based on records to the best bulls based on the daughters of the record of the daughters of those bulls. Then the young calf was grown up, and until it was ready to, you could collect semen from this male animal. And once you had the semen, you were inseminating around 1,000 cows and waited for the for the performance of about 50 daughters. And because it's similar, it's a dairy breed in Austria, so you have to wait to find out which is the best one in this way, that you wait after five years or whatever it takes the cows to get, or the daughters to get old enough to produce milk, and based on that one, then bulls are used very heavily. And so the top bull here in Austria, for example, he produced the AI companies sold 500,000 doses of semen of this particular bull, which of course leads into other issues related to inbreeding, but I will not touch this one here. So, and what is being done to find out what are the best animals is essentially we use a methodology that's called BLUP, linear unbiased prediction. So it's just a statistical method, and essentially the only thing that this tool does, it solves systems of equations. So the phenotypes are the traits, and you can also do this one in multiple dimensions. So you can do multiple trait analysis all at the same time. So the phenotypes are the traits, and the environmental factors like herd or year or whatever it can be. So many of them are covariates. And then the genetic value of the animal is also an effect to solve for. And what those people have come up with is using not only the information of the animal itself, because we are interested, our key interest is the milk production of a bull, or ability of a bull to transmit genes for milk production. So essentially it's working very much via relatedness. So pedigree relatedness, that's what has been done. You set up a relationship matrix and implement this one into this prediction equations to link observations. So with genomic selection, things have changed because very obviously you could see, I feel it's obvious, if you had this previous system, you had to wait a long time before you made a final decision which bulls to use heavily. And if genomics would give you this information earlier, so that you can even from a young calf already know, well, this is a good one or this is a bad one, and using genomic information for this one, this would reduce generation interval and consequently genetic gain. And so the key is actually the short generation interval that's increasing the genetic gain. And so genomic selection, and now we are talking about single-step genomic selection, is being used very, very heavily in dairy cattle breeding. So, and right now, getting 50,000 markers across the genome is costing in the range of $20 or 20 euros. And what people like me are doing is adding to the BLUP equations that we had before, where we had phenotypes and pedigrees. Now we just add the genotypes to this system. I will not go into detail, but it is all mathematically very complex, but it's doable, and people have found good solutions for this one. So, but right now we combine pedigree relatedness with genomic similarity to solve the system of equations where we get the breeding values of the animals as a solution. And that's what we want, because we want to find which are the best ones, which are then used as breeding bulls or mothers of the next generation of cows. So just to tell a bit about breeding value estimation or genomic selection in Austria, we do have a joint system together with Germany and Czech Republic, because Fleckviehs or Simmentals is our main breed, and so about 80%, as I told, 80% of the animals in Austria are Fleckvieh or Simmental. It's just two synonymous words, sorry for this one. And there are about 400,000 cows being recorded in Austria, but there's more than 1 million recorded in Germany, and in Czech Republic it's a few 100,000, I think 100,000 or 200,000. So, but putting things together and combining information about records, phenotypes and genotypes and pedigrees is just improving the system a lot. And so they have a group of scientists or well, lab people, lab in terms of biotech lab people who would work on this one, and the key staff in Austria, they have all been my PhD students, and the Austrian team in this system is responsible for functional traits. That's how they split up their work. And just to give you an idea again about numbers, so it's roughly, I think it's a bit less than 2 million cows being currently under recording for Simmental, but we also use old data up back to the 1980s for records, because our animals are related to each other, so there is also extra information in the cows that were bred or lived in the 1980s or 1990s. Of course, much less than nearer neighbors or nearer relatives, but there is some information there. So essentially, the system of equations has about 20 million unknowns, and it's solved at the same time. And also right now into the system, if you think about like 50,000 markers of the genotyped animals, you come up to 50 billion individual genotype values going into this system. And currently about 400,000 of those, well, animals, current population is 2 million, so quite a large proportion of the animals is actually being genotyped. And farmers are willing to pay those 20 euros for getting extra information for their cow, so that they know also for their cow, is it genomically high or low. So they are willing to pay for this one. So right now every 10th newborn calf is genotyped. And of course the very young male calves that are being selected, they go through a process. There's a lot of male calves that you don't know the milk production, you're not interested in the milk production of the male calf, but for being selected as a breeding bull, you're highly interested. So this is all kind of working extremely well, according to my opinion. I tried, because you are mostly human geneticists, or quite a few of you are human genetics. So my question I put here, and I'm very bold on this one, and we can discuss this one, is that I think that genomic selection in livestock does set an example for human personalized preventive medicine. Because we are recording phenotypes, and if you would have, I don't know, medical centers who record blood pressure, for example, or triglyceride values, or whatever you would measure, it would go very similar way actually. And so livestock people have actually, so the human geneticist people have been aware that this is existing in livestock, and they are also interested in our kind of expertise and collaboration. So talking about lab, so my lab is very close linked to the Austrian cattle and pig breeders, which is coming, I have to be honest, also from my background, because it was always my key incentive as being a professor to improve the livestock populations genetically, mostly in cattle, but also in pigs. So I have been developing blood procedures for new traits, like persistency of lactation, longevity. Then also we have been asked as a group by the cattle breeders in Austria, because this is by law, animal breeding is regional responsibility. But then the cattle breeders came to us, and this is, I think, the only time, at least I wonder, that they came to us and said, could you help us setting up a joint breeding program? And that's what we did around 2000, and as indicated, coming back from Australia, I brought back genomic selection, and we set up a project on this one. And again, this is coming back to what Natalia told, because I've been working so long in this field, and I have earned trust from the breeding associations. So they think that what we are doing is right. So this was, even when I brought back genomic selection it was not, had not been used routinely at all anywhere at that time. They said, well, we will invest into this one, we will genotype 2,000 bulls because we trust you that this will work out. And so essentially I think I have helped turning animal breeding from art to science to quite a large part in Austria. So, but what we are, or what my group is we are kind of a pure bioinformatics lab. So we have not invested in a genotyping lab at all. Also thinking about what has been talked here, maybe it was not a bad idea because the genotyping equipment is super expensive and it's changing so quickly. So collaborating or even buying in genotypes might be a better option than having your own lab. I'm not 100% sure. Maybe at least having a lab where we could extract DNA would be a good idea, actually. But yeah, it's a way of thinking how to go. Well, and essentially, so what we are doing is we are solving equations. This is our kind of key task or key expertise. And so we have a group of about 4 permanent staff and about 5 to 10 doctoral students. And I personally, I'm so keen on science that I never went up into any higher administrative position. I didn't want to do this one because then I thought I would have to leave what I love to do. And also I think this group size, you can discuss this one, this could have been a bit bigger. But well, it's also a nice size actually. So we have Molana here, for example, I love to work with him, and so it's just a good situation that we are in. And again, yeah, so what we have as this bioinformatics lab, we have access to huge amounts of data, but as was also told yesterday, there's much more data around than can be analyzed sensibly anyway right now, but we are having this link to the breeding associations. We have this very good link to very good phenotypes as well, not only genotypes. And so we work mostly with SNP chip data, whole genome sequence. Molana will give a presentation tomorrow on this one. And we also use mtDNA sequence from time to time. So my kind of statement here is try to be expert in some fields, not too many, and have a group that is reasonable but not too large, but also, of course not too small either. So the field that we are good at is analysis of population structure and admixture. Collaboration with Natalia's group is just great here. So some of the graphs you saw, we did together. Then my personal interest is very much into inbreeding and inbreeding repression, as well as crossbreeding and heterosis. And so we work in this field. I think I will not go into this one much. It's just, well, because it's just one small aspect that was my personal interest, but I think I will not use time on this one. So what we can't do right now is we can't, so it would be good to have a lab sometimes to be able to quickly sequence a small part of the genome following a signal, things like this. We can't do right now expression analysis, RNA-seq. We can collaborate and we can with institutions who can do this one. But of course, particularly for the first part, I sometimes do feel a bit unhappy not to have this one, but again, it's all about choices actually. You make this choice, you don't have another choice and things like this. So the solution to that one is just, well, if we don't have it, so we collaborate and we show that we are experts in analyzing data. So of course also like institutions are happy to collaborate with us. And now I have quite a long section on collaboration because I think this is super important in running a lab actually. So because thinking together and solving a problem, none of us is smart enough to know everything. So as soon as we are collaborating, we are having new ideas together, and so it's just incredibly better. And particularly what I feel also, like people from different institutions and more so from different countries, they have just different types of experience and they think differently. Overall, we have the same kind of network and thoughts, but still the way you have been taught to thinking is still giving you extra advantage, and I love this one a lot. I will very briefly go through, well, 6 types of collaborations that I had over my career. I started in Australia, where I did a postdoc directly after my PhD, working on optimum design of cross-breeding experiments. Then I went back there because it was really nice to work on gametic effects in livestock breeding. And as indicated, I was invited to be visiting professor at the University of Sydney for a full year, where we worked on genomic selection, when genomic selection was not yet out. So the concept was known, but nobody had data, so they were among the first ones to have enough data to see whether they can prove this concept. And that's also another point down here, collaboration with other countries is not only me going there, but in all cases, my students and my postdocs also went there at different points in time. They had their own projects. But again, just thinking about working together is you establish you establish trust between institutions, and having on this based on this trust, then students should travel. Well, for Europe, I don't have so much. Well, being in European Union, we have a lot of collaborations, so it's going on all the time. And overall, the network of collaboration is reasonable in European Union. Also, European projects, they're sometimes too big and things like this, but overall, the network of scientists in my field is very, very useful. And, well, also very on par, so it's kind of nobody thinks who is the best, something like this. Then I have a personal mission since about 15 years. I go to Africa a lot, and I go there personally a lot for this one, because, well, essentially, people always claimed, well, cattle breeding is nice, but you can't do it in Africa because you can't establish a recording system, on-farm recording system. And I always thought, why should that not be possible? And so I try to work on this one very, very heavily for quite a long time now already, establishing what we call community-based breeding programs, which is kind of on-farm recording and using this information. So, and I had many PhD students with me at BOCO. I had also a lot of Austrian students, but I think it's already half-half right now, half African and half Austrian. Or again, so just this is a paper that was published this year. Another one. In Brazil, again this came up because a colleague from Brazil, whom I didn't know, he found that I would be a person probably collaborating with, would be beneficial for him or his group in genomic selection. So he invited me to come over to help them with implement genomic selection with the Nelore and Gyr cattle breeds. There's lots of Nelore in Brazil. It's more than 100 million of them. And they're a beef breed. There will be a slide on this one. And also, like then, I had a 6-month contract with the Brazilian government as a special researcher, special visiting researcher on this one. And so here we have the photos up there. This is the beef cattle called Nelore, and right down it's the dual-purpose dairy cattle called Gyr. And of course, Brazil has a lot of cattle, and particularly a lot of beef cattle actually. Okay, now come to the best. So I can't remember the year. Sasha, was it 2012 already, or was it later? I forgot. 14. So sorry, this number is wrong up there. Well, and essentially, Natalia had the idea, as she indicated, also discussing with Gottfried Brehm, who would be the person to collaborate, or the group to collaborate with, related to SNP chip data, genomic data, and he suggested me. And since then, Sasha Sermiagin and all, also the other colleagues, they have been at BOCO repeatedly, And even if they are not there, so right now, this is because I'm writing down there, I'm writing collaboration at its best. What is still missing for the very best would be to have long-term visits, both directions. So we did not have this one so far, but we started trying to do this one. We tried starting doing this one with 2019 when Corona was already on the horizon. But I think this will make it to the best by also having longer-term exchange visits there as well. So, but we did a lot of publications, and the three of them who are here, they're just doing a very, very good job. All of them, Veronica, Arsen, and Sasha. And it's I personally love collaboration with you, and I love to be here for this particular meeting here as well. So this was the very last publication in Frontiers in Genetics. Please think about publishing in Frontiers in Genetics. It's a cool media. I'm the chief editor for livestock genomics in this one. So think about this one, because they have all fields of genetics, and I think it's a good type of journal. And our next project will be, Natalia already showed that they have established this ancient DNA lab here, and we have my predecessor as a professor. He collected samples from about 1900 to 1930 in Austria and going east. So we have a lot of samples. So there's a big collection there, and we will use this collection and link it to the collection that is to the collections actually that are here. So this is one of a project that we are about to submit jointly. We wanted to submit to the Austrian Science Foundation and the Russian Science Foundation, but the next joint call is only in 2 years, so we don't know what we will either. Well, probably we will submit to the Austrian Science Foundation with collaboration from Russia. Okay, so my kind of conclusions on this one are that research in livestock and breeding and genetics is currently at an all-time high, and high throughput genomic data at a low price have made this possible. So it's super cool what we can do. Again, as indicated, it is important to use the data, not only to collect them, but use the data. And so we try to work on this one. And so my view of best practice of such a lab is to concentrate. So this is, but this is just because it's me. I did not want, like Natalia, she has had a bigger vision that goes further, but my vision was just doing a good job in my own group, so to speak. Collaboration, international collaboration is just cool and very, very important, because you get different thoughts and different frames of mind, and together. And well, I did not talk about this one, but of course it's also, as a leader of a lab, you have to think about your group of people. And again, I have Superwoman for this one. I talked with my colleague, we talk repeatedly, and she is doing this one to a very, very good extent. Thank you very much to everybody.
Hans, thank you very much for inspirational talk. You're exactly in the line what we wanted. So any questions or comments? Because I have some, and I can probably start with. Hans, I have a question. So the selection, this is S&P based. It's one field, so genomic information is enough. So it's just order to make a chips to select best among animal. But when we are coming to the era of gene editing, when you want to modify, not to make a marker to select, but just to modify. But the question, do you have enough information on cow, for example, or maybe better implementation of transcriptomics data, or better annotation of the genomes of these cattle breeds are essential? So how do you feel? What is the direction? So do we need to integrate more gene expression data, or it's just enough? So what is the vision in this area?
Okay. So I originally had a plan to have like a separate section on gene editing, my view, but I took it out just because of time reasons. Again, at the breeder's perspective, many cattle or livestock breeders would think that gene editing will not advance enough because we don't have big enough targets, in terms of effect actually. So people are doing analysis, and particularly, several people in Europe saying, well, you can also do like a multi-editing with many, and then you can improve. Personally, I'm not very convinced. I have, again, for my African case, there is a single gene that's called, SLIC, that does has a strong effect on heat tolerance of cattle. So that one I would edit immediately, but I'm not sure about this multi with many different edits at the same time. Also because I don't know too much about how potential interaction effects would work. But yeah, I would guess if you would go this way, then you need to do expression analysis.
Because this is the area we're planning to collaborate with Natalia, actually. Okay, any other questions or comments? Well, I have another one, Hans, actually. I'm sorry, I take my role asking questions. I wanted to ask Natalia, actually, but because of time constraint. So you collaborate with Natalia for like years, like me, so maybe seven, 8 years, so it'll be soon.
Yeah.
So if you can jump back in time, so in a line of collaboration, what would you change initially, or update, or make it in a different way, make it, I think, more intensive visits, or some area to be started earlier? So kind of,
two things actually. First of all, we have not traveling enough to Russia, so you have come to us, and just again we talked a lot, so we did have a lot of communication, and we also talk a lot also outside of the visits, of course, but still, because I'm doing this one, for example, with Africa, other places, I go there and better understand what's happening. So I think that's what I would like to do very soon. Like also just having a 2-3 months visit myself, and also send students here for half a year, something like this. So this is what is missing right now. And I can do this one because, again, it's boiling down to trust. Because if you have, the collaboration is as with association with breeding associations, is very much related how much do you get on with the person, or are you comfortable with the team? And I'm very happy with you, with collaborating with you.
Okay, thank you. So, any other comments or questions? If not, okay, let's thank Hans again. So please use your chance to talk with him because it's quite inspirational. And Hans, I think we will welcome you definitely for this PR section we have tomorrow. Because as an editor of a journal, it will be quite interesting for us to see vision from Austria on this promotion of research results. So please stay around.